Okay, so far we have learned about Gregor Mendel and his pea plants and the idea that uh, genes come in two different forms called alleles. And these two different forms come in two sort of variations where one is dominant and the other is recessive. And what that means, remember, is that the dominant one will mask or hide the recessive one. And we've seen all the different combinations that we can do. We've done the Punnett squares. And what Mendel did, of course, was he bred the yellow and green pea plants, and he kept track of all the different possible combinations of offspring and looked at the ratios that they were coming in. And he was able to produce a theory of these alleles. He called them factors, of course, and the fact that one was dominant and recessive, and he got results that corresponded to his actual pea plant experiments. And that's how science basically works, right? We have an idea and then we check it against reality. And if it works or matches what we observe, it tells us our idea is a good one. But there were a few things that Mendel was not aware of, or that he couldn't explain. And it took a while for us to understand all of the details. So now what we're going to do is we're going to look at something that's a little bit different. We're going to look at a situation that was going to be called incomplete dominance. And this is something that occurs in some genes. Rather than having a dominant and recessive gene, you have a situation where neither one is completely dominant. So neither allele is completely dominant over the other. So what does this mean? Well, it means that both alleles will be sort of blended together. Blend together to produce a third phenotype. Remember, the phenotype is what you look like. So there'd be a third option. So before, we had yellow and green pea plants, and we had babies that were either yellow or green. But now we're going to look at a situation where the alleles actually blend. So we're going to look at the example of the snapdragon, which is a flower. The snapdragon shows incomplete dominance in the allele for color. So the color of a snapdragon is determined by two alleles. One is red and one is white. But because they're not dominant or recessive over each other and they kind of blend, we have to use a different notation. We can't use a capital and small letter because that would indicate one is recessive. These are equally powerful alleles, or at least they are sort of not masking each other as much. So the color of a snapdragon when it's red has to be represented by the allele C for color, capital C, and we'll put a little capital R up above to show red. The other color that you can get for a snapdragon is white. And so we use the allele color and a W for that. So we're not using any small letters at all, because this allele for red blends with this allele. It doesn't totally mask it or hide it. So here's what happens when you do a cross with what we call incomplete dominant alleles. If we take a red snapdragon and we cross it with a white snapdragon, here's what we get. Let's do the genotype, or sorry, the Punnett square. A red has CR alleles only, right? Its genotype from the mother, or its genotype mother or father, would be CR, like this, right? So homozygous red. But the only allele it could make is this one. So really, I don't even need the bottom of this Punnett square, do I? Because there's only one allele that can be made. Let me just redraw that Punnett square because there's no point. The white one is CWCW, homozygous white. And we'll see in a minute how I know they're homozygous. They can't be heterozygous, because if they were, then one would mask the other, right? Okay, so the only possible allele we can get from the white flower is this one. No matter how many ways you look at it, all the sperms and eggs will be C with a W. Now when you cross them, you get this situation, a CR and a CW. But if one is not dominant over the other, what does this produce? This produces a third phenotype, a brand new phenotype, which is a combination of the red and white, which is pink. So in other words, instead of getting just red or white, it's possible to get a blend in an incomplete dominance situation. And so then what happens is, if we look at the possible genotypes, there's only 3 possible genotypes available. If you're a red flower, you must be this. If you are a white flower, you must be this, red, white. And if you are a combination of those two, then you will be a pink flower. See, the third possible phenotype. And that's it. Those are the only combinations. So let's just go through the cross again. We've already did the first cross. We saw that red crossed with white. And because red was CR, I'm just summarizing again what we just did. Cross with C white, color white. The only possible gametes, if we think about the gametes, the only possible gametes from the first one are this one, and the only possible gametes from the second one is that one. And when we cross them, it's a very simple cross, and it always produces pink individuals. 100%. 100% of these individuals will be pink. It's impossible for these two flowers to cross and get a red flower or a white flower, because the only combination is an R and a W. So 100% pink flowers. But things get a little weird if we do the next generation, the F2 generation. We take these pink flowers and we cross them. What would happen if we did a cross with one of the pink flowers from that generation with another pink flower? Let's cross 2 pinks. The pinks have genotypes C, R, C, W. And when we look at the possible gametes that could be formed from these sperms and eggs that could be formed, we see that this one could produce 2 possible gametes. The CR or the CW could be in the sperm of the egg. And the same is true for this one, the CR or the CW. So there are 2 gametes now. And when we do our Punnett square, we see that we have to do the old-fashioned one here, the one we did before. There are 2 possible gametes for mom, these 2, and 2 possible gametes for dad, these 2. Now when we cross them, we get a whole variety of things. 2 CRs, here's a CR and a CW. Here's a CW and a CR, which is kind of the same thing, just backwards. And here's 2 CWs. So now look at the ratios when you cross the pink flowers. We have 1 out of 4 CR, CR. And what color would that be? Red. So 2 pink flowers can actually make a red flower. Then I have, if I look at these 2, they're essentially the same, just reversed. That's 2 out of 4 CR, CW, which are pink flowers. And the last one, right here, CW, CW, 1 out of 4, which is a white flower. So, in the first cross, a red and a white flower produce only pink. But because the pink are heterozygous, they are a blend of the two alleles. When they cross, those alleles mix up in 4 different ways. Well, 3 different ways, really, right? 3 different ways. 25% chance of getting a red flower, 50% chance, 2 out of 4, of getting a pink flower, and a 25% chance of getting a white flower. So now all the colors kind of come back in the next generation. So that's how we deal with incomplete dominant. That codominant comes later. Incomplete dominant alleles. We use a capital letter for the allele, and we put the 2 options as capital superscripts, and whenever we have 2 of them occurring, we get what's called the blended phenotype. Red and white would produce pink. And that's how we do it. And so you're now going to do an assignment where you're going to try to figure out some genotype ratios and phenotype ratios using this concept.